Here. It is truly a singular pleasure for me to introduce Ed Vogel to you. So Ed was the very first graduate student I ever recruited, and I probably should have just stopped after that. So
it's ambiguous.
Yeah. It's like, you know, in a letter of recommendation saying you'd be very lucky to get him to work for you. Think about it. Ed came to my lab at the University of Iowa in 1995, and he did a little first-year project, as students do, on the attentional blink and the N400 that was published shortly thereafter in Nature. So that was his first-year project. And then in his second year, he started working on visual working memory using change detection tasks and did a few experiments that ended up in Nature. That was an auspicious beginning. Now, it turns out that there wasn't a Nature paper every single year, but there were many, many great papers. Ed did fabulous research as a graduate student that really led to a lot of what I did after he left the lab. This is my model for graduate training. You bring them in, you show them a few things, then by the time they leave, you know, you're following them along. So after he got his PhD in the year 2000, Ed did a postdoc at UCSD working with Marty Serino and with Steve Hilliard. And Steve Hilliard was my graduate mentor, so this was a lot like having your child go live with their grandparents in the old country. Now, Ed didn't spend too much time there because he very shortly thereafter got scooped up by the University of Oregon, who hired him as an assistant professor. So in 2001, right, you moved to Eugene, and he had this strange overlap period where he was both still a postdoctoral scholar at UCSD and an assistant professor at Oregon. I think that's called double dipping.
It was great.
About the first thing he did when he got to Oregon was start to use ERPs to look at visual working memory, and he discovered this brain activity that's related to the maintenance of information in visual working memory, and started looking at individual differences and showing that you can actually, I will not take his entire time, by the way, showing that you can actually look at individual differences in working memory capacity and relate them into individual differences in brain function. And this turned into, you got it, a Nature paper. And I was so happy about this because basically what it did was it made it clear that my letters of recommendation for him had not been a lie. And you know, when your students leave the nest and they go off, you never really know, can they really succeed by themselves? Well, this made it clear that he could succeed, as did the follow-up Nature paper the next year. And there have been, you know, again, a succession of great papers after that. So it's really great to have Ed back, and I'm very interested to hear what he says. So Ed today will be speaking on individual differences in disengaging from attentional capture.
Well, that's a really difficult introduction to follow. I do know of other journals other than Nature. I've heard of them. Well, although in grad school, I think that's, Steve just told us that was the only journal that existed, and so that was pretty much all we had to shoot for. Well, it's a real pleasure to be here. This is my first time in Davis, and it's kind of this strange homecoming because I have so many friends here, but I've never been here before, so it's like this familiar but new and very exciting to see everyone here. So today I'm going to talk a bit about work that we've been doing in my lab looking at individual differences, as Steve mentioned. But before I begin that, I want to kind of sort of draw your attention to my co-author here, my very talented, very brilliant grad student, Keisuke Fukuda, who couldn't be here, but I'll just put a picture up of him in his happiest moment when he caught a steelhead on the Columbia. So he's really happy when he's collecting data or he's catching fish. Those are pretty much his primary passions in life, I think. So I'm going to talk a bit about work that Keisuke, we call him K, by the way, and he studies K. We'll talk about that in a second. But the work that we've been doing recently to characterize how people vary in their attentional abilities, in particular looking at variability in how they perform memory tasks and how that could really tell us something about sort of broad aspects of their cognitive abilities, and also look at the neurophysiology that's associated with it. So first I'll start off with work that that Steve Luck and I began when I was a grad student in his lab at Iowa, where we were interested in trying to assess the capacity for holding multiple items in working memory at the same time. So how many things can you think about, even when you kind of simplify what they're asked to think about, how many separate objects can they hold in memory at the same time? And we did that using a simple paradigm that we refer to as the change detection paradigm, where you flash an array of simple objects like colored squares for a brief amount of time and ask subjects to try to remember those as best they can. You take them away for about a second or so while they're remembering those items. And then after a second or so, you flash up some colors on the screen, and on half of the trials, the colors they see are exactly the same as the ones they saw originally. And on the other half of trials, we change the color of one of the objects. And many of you are probably pretty familiar with this general paradigm, but one basic aspect of these types of tasks is that when we introduce a change, it's generally a really large change. So it's not just a really subtle change from red to slightly less red. Here, these are really large salient changes going from, say, yellow to purple or blue to green or something like that. Something that would be a normally very easy salient change to detect, so the performance wasn't limited based on their perceptual acuity or the resolution of the representation, but really more about whether they just had some information about what those objects were. And the way we measured memory capacity in these tasks is simply by varying how many items were in the display. And you can imagine that if there was only a single item in the display, it'd be a very trivial task. If it was red to red, you're like, okay, that's same, or red to green, and that's really easy to tell those different. But then you add one more item and another item and another item, and you find the point at which they can no longer detect these really large salient changes anymore. And what we and others have found over the years with tasks like this, with large changes, that you find that on average people can remember somewhere around 3 or 4 items. And that's typically what we have found, and that's kind of what we focused on back when I was in Steve's lab. And one thing that's kind of interesting about this is you can come up with an estimate of someone's memory capacity, and I won't bore you too much with the exact formula. It's pretty much just a way of taking that change detection performance and just coming up with just an estimate of what their average capacity is. And it turns out that in these types of tasks, it appears that there's a developmental time course in terms of your capacity for doing these types of working memory tasks. So for example, most of the time we focus on sort of healthy young adults that are, say, 18 to 45. But what you see is that in these types of tasks, that if you have children perform the task, you tend to get considerably lower performance, as well as much older adults. You actually see that performance actually starts to decline again after 65 or so. In addition to that, you can also see reductions in performance in this task in a number of different populations that have either neurological or psychological deficits. You can have individuals with schizophrenia tend to show a really dramatic reduction in performance on this task. This is like work that Steve and Jim Gold have been doing over the years. Also in individuals with Parkinson's disease. And you can see what's kind of interesting is that often these are patients that have problems perhaps with relation to regulating dopamine function. So I won't have a chance to really talk about that, but you do see really dramatic declines in performance on these very simple tasks in a number of different patient populations. Now, what I'm going to focus on today is within the healthy young adult group, even though you typically get a capacity estimate of somewhere around 3 or 4, there actually are really large and reliable differences across individuals in performance on these tasks. And I'm going to argue that those can actually be really a meaningful way of getting a sense of what their sort of overall cognitive ability is, and at least in a number of different contexts. So what I'm showing you here are 170 healthy undergraduates at the University of Oregon, where we had them perform this colored square change detection task. And we're just simply binning them on the basis of what their memory capacity estimate is, or we call that K. And what you can see when you look at this distribution here is that, well, most people cluster somewhere right around 3.5 objects. You get an average about 3.8 in this particular sample. But you also see that there's actually a pretty substantial spread in K estimates or memory capacity performance. You have some people that are on the really high end that are somewhere in the vicinity of 5.5 objects or more. And you also have people that almost seem like they have sort of an impossibly small memory capacity. You can have individuals that have 1.5, even lower than that. Now, an interesting aspect of this is that While there is some variability from day to day for a given individual, it actually tends to be a very reliable measure of their memory capacity. So in these studies, we've actually had them come in and perform this change detection task. It takes about 20 minutes. It's a pretty quick procedure. And then had them come back 6 months later and perform the task again. And we find a test-retest correlation in this measure of 0.82. We generally get this, so we have a really high level of reliability. It's actually a fairly stable trait of the individual. You also find that an individual's memory capacity, as measured this way, tends to co-vary with a wide range of sort of high-level aptitude measures of the individual. For example, it predicts, it has a nice strong positive correlation with Stanford Binet IQ, for example. And so one thing I'll sort of emphasize here, and this maybe I think will help to kind of frame some of the work that we'll be talking about in a few moments, is that in these types of tasks, when I'm showing you this type of data, where we're measuring memory capacity, we're estimating their capacity on the basis of trials in which we gave them a lot of objects on the screen. So for example, these are typically from trials where they had 8 items on the screen. And essentially the idea is that, well, if you give them a small number of items, say 4 items, which is typically right around capacity limits in general, you see much more restricted range. There isn't nearly as much spread. And one possibility is that, well, it might be that some subjects have a higher capacity than 4, and so you're underestimating them there, and that might be one reason for the spread. On the other hand, another option, and that will kind of be sort of endorsing, is that really when you go to these large array sizes, you're not just measuring how many things they can hold in memory, but you're also measuring how they respond to being overloaded and how they can deal with having a huge number of items that come on and how they can control their attention under those circumstances. Now, what I'll show you just really briefly is we can even, for those who are familiar with other types of high-level cognitive testing like intelligence testing, fluid intelligence correlates really nicely with this measure. So this is performance on the Raven's progressive matrices, which is a measure of abstract reasoning, visual reasoning. And you see nice positive correlations with memory capacity just with colored squares. So just by measuring this, getting this K measure in about 20 minutes with colored squares, we can explain about 25% of the variance in measures of fluid intelligence. And we've done more extensive work using latent variable approaches and also structural equation modeling, where actually once we really kind of have multiple measures and have these more sophisticated approaches, we can actually explain upwards of 35 to 40% of variance in fluid intelligence. So it appears that something as simple as this colored square task that we kind of stumbled on when I was in grad school ends up kind of explaining a wide range of variance in fairly high level measures of cognitive performance. So now I'll shift gears a little bit and talk about some work that I think Steve alluded to, some of the neural approaches that we've been taking to measuring information that's being stored in visual working memory as well as individual differences. And in particular, we use event-related potentials by measuring electrical activity at the scalp. And we've focused on this particular component that we've referred to as the contralateral delay activity, which I would argue gives us this online measure of how many objects are currently being held in working memory for a given individual on a given trial. So here's essentially how we go about doing that, and it's essentially the same basic task as what I showed before with one slight twist, and that is the subject is fixating, and we're measuring, they're making sure and forcing fixation, we're measuring where their eyes are, and they're holding fixation at the center, and they do that all the way through. And essentially, we give them a memory array, just like I showed you before, and they have to do the same type of change detection task. With the difference is that while they're fixating, we give them half the objects in one visual field and half the objects in the other hemifield. And we ask them at the beginning of the trial to only remember the objects on one side or the other side. So we'll give them a little arrow cue that says, okay, now on this trial, just remember the colored squares on the left. So on this one, they would just remember these objects, and then at the test array, they would make a same or different response for those objects on that same side. And the reason why we do that by having a bilateral display like that is that we can exploit the fact that the visual system is primarily organized in a contralateral fashion. And what we can do is it allows us to separate out the visual evoked response in terms of the perceptual response to stimuli coming on the screen, and the brain responds to that. And we can separate that perceptual response from the attention and memory to one side of the display. That will be unilateral. And so what we can do is we can look at the electrodes, the activity over electrodes that were contralateral or ipsilateral with respect to the side of the display they were asked to remember for that trial. And what we find over the posterior electrode site, so right around here or so, we find this sustained negativity over contralateral electrodes with respect to the side of the display they were attending. So I'll just kind of walk you through this real quick. This vertical bar is when the onset of the memory appears. This red bar here, that's the 100 milliseconds when this is actually visible. And the bulk of the rest of this waveform is actually when they're just staring at the screen and remembering those colors. And what you can see is that the purple line is the contralateral activity. What we can see is that about 200 to 250 milliseconds after the onset of the memory array, you see this large negative wave, and these plots, negative is up, which is the way God intended it, is my understanding.
Ever heard of a Cartesian coordinate system?
I've never met him.
I so agree with you.
Yes! Okay, so you have negative is up, and you see this sustained contralateral negativity that persists all throughout the entire retention period until the test array arrives here at the end of the trial. And we refer to this as the contralateral delay activity. And we can isolate this activity simply by doing a subtraction between the contralateral activity and the ipsilateral activity. And that's what we refer to as the contralateral delay activity because it's reminiscent of delay activity that's been observed in monkey single unit recordings when monkeys are performing match to sample tasks. You see the sustained activity during the delay period. And a really exciting aspect, at least for us, of the CDA is that the amplitude of this activity scales as a function of the number of items that are currently being remembered on that trial. So I'm showing you the difference waves, the contralateral minus ipsilateral activity. So these are the CDA waves. And what you see is that if they're asked to only remember a single item, you see a low amplitude. But if you go to 2 items, you see a significantly greater amplitude. You go to 3 items, it gets larger again. But when you go to 4 items, it doesn't really rise up much more. And at first we were kind of puzzled by that, but then we realized, well, wait, that's right around what the typical capacity limit is in these types of tasks, at least what we were estimating based on behavior. And so we went back and looked at a wide range of set sizes. And here I'm showing you the mean amplitude of this activity as a function of array size. And what you see is that you get this increasing function from 1 to 2 to 3, and then after 3, it essentially reaches an asymptote. You see a little bit of a decline as you go to larger array sizes, but I'll talk about that in a moment. But it pretty much reaches an asymptote, doesn't continue to get larger after about 3 items. It pretty much holds steady. And which would fit with the idea that it's, the visual working memory has sort of reached its maximum, and even though you've presented 8 items, you can't hold any more than, say, 3 or 4, and so that's about, you've sort of hit your general limit. Another aspect of this that we found particularly compelling was that, well, one possibility is that the reason the amplitude increases is simply because the task becomes harder. You go from 1 item to 2 items, the task is harder. Maybe this activity is just about how much effort or energy the subject has to expend to do the task. Well, one nice thing about the fact that we find an asymptote is that it helps argue against that possibility, and that is, well, set size 6 or 10 is much, much harder than set size 2 or set size 3, and yet you don't see this continuing increase in activity. So it's not just simply the difficulty or arousal that the subject is engaged in, it fits better with the general capacity limit. What this red line reflects is the mean capacity estimate based off of behavioral performance in this particular task. And you can see that it's, in this particular sample, it was about 2.8, which is right around where this component reaches its asymptote. In fact, what we found, got us really sort of pulled me into the world of looking at individual differences was that we found that the individual's memory capacity derived from their behavioral performance was very nicely predicted by this particular component, and specifically looking at the change in amplitude, the rise in amplitude from 2 items to 4 items. If we look at the rise in amplitude from 2 items to 4 items, we see a really beautiful correlation with an individual's memory capacity based off of their behavioral performance. Now, originally we viewed this as something to be that the high capacity subjects maybe had a higher point at which they reach asymptote, where the low capacity subjects reach a lower asymptote. But in a moment I'll show you some more recent work that we've done, which I think changes our view a little bit on that, but I think still sort of fits within the general idea. But the real key here is that we see a really strong correspondence between individual variation in behavioral performance and activity that we can measure at the scalp. We also have a nice measure of how many objects they're currently holding in memory in a given situation. Now, people have done neuroimaging studies very similar to this, and one possible locus, or at least partial locus of this particular activity, a good candidate appears to be the intraparietal sulcus. When subjects perform similar types of tasks to what we use, but do that in the scanner, you can see that the IPS shows a similar kind of profile to what we find with the CDA, where you see bold activation that's modest for one item, raises for two, raises again for three, and then reaches some asymptote. And in later work by Todd and Marois, they even found that the exact point at which it reached asymptote was also sensitive to individual differences in behavioral performance on the task. Now, I wouldn't say that the IPS is the sole locus of this activity. Anyone that does ERPs generally knows that it's very rare you find a big component like this that's got a single source, but IPS seems at least to be a likely contributor to this, this activity that we're seeing. Now, one thing that we've been really focused on, and the rest of my talk will really kind of emphasize some of these basic questions, and I can't say that we've completely answered them, but I think we've got some compelling data. We're really interested in what's the difference between a high capacity individual and a low capacity individual, particularly now that we have an idea that these things are related to high level cognitive performance. So we want to get a better handle on what the difference between a high and low capacity subject is. And one idea, and this has been sort of a dominant view for over 100 years of looking at individual differences in working memory performance, is that, well, the difference between a high capacity or a low capacity individual is about how much storage space they have. High capacity subjects may have more slots or more representations that can have active simultaneously in memory. They just have more RAM, to use a computer metaphor, versus the low capacity subjects that may have a lot less storage space. In contrast, we've been sort of examining an alternative model that suggests that perhaps everyone has roughly the same amount of storage space. That might actually be relatively fixed across individuals, and that really the differences between high and low capacity subjects, it really is dependent upon how well they control what information gets stored in memory, particularly in situations when you give them more items than they could possibly store. And so it might be that the low capacity subjects really have a poor control over what they store, and as a consequence, when you give them too much information, they might try to store all of it, overload capacity, and performance really suffers as a consequence. So I'm going to talk a bit about some experiments we've been doing recently that try to distinguish between a few of these models and at least provide some evidence in favor of this hypothesis. The first thing that I'm going to show is an experiment we did very recently, actually, and it's kind of a throwback experiment because it's almost identical to the one I just showed you before looking at the CDA, except in this one we were going back and doing that same basic experiment again, except in this one, in the previous one we did 3 different experiments, one that had tested set sizes 1, 2, 3, 4, another one that was 2, 4, 6, another one that was 2, 4, 8, 10, but we never really had a nice continuous measure of all the different set sizes in between. And really, we wanted to sort of really test further this idea of, do high capacity subjects have more slots than low capacity subjects? So, for example, we consistently get individuals who score really high on these performance tasks. They'll get a K of 5.5 or better consistently. And one idea is that, well, if they really do have 5 active representations simultaneously in memory, in working memory, well then you would expect that if we measured their CDA, just those individuals alone with a K of 5, well then you would expect that the function would look like, well, it would rise up to 5 and reach asymptote. On the flip side of that, you could say, well, if you have an individual that has a capacity of 2, a K of 2, well, then you would expect that they would only rise up to set size 2 and they would reach asymptote. And our previous work had suggested that to some degree. But we wanted to go back and run a really large sample, and so at least for ERPs it was large. We ran 65 subjects, which is a lot of subjects. And plus, we did all 1 through 8 set sizes. So these were long experiments, and it was a lot of people. But I think it was worthwhile. So what I'm showing you here is the CDA amplitude for the whole population, the whole group of 65 subjects. And what you can see is that, just like I showed before, you see a rise up to 3 and an asymptote, and you even see a little bit of a tail off when you get to the really high array sizes. Then what we wanted to do, because we had such a large sample size, one main reason we did that was that we wanted a nice large population of individuals that had a K of 5 or better. We also had so we had 15 of those, 15 subjects that had K of 5 or better. We also had 15 that had a K of 2 or less. So we could have an extreme groups design where we had individuals that had this really high capacity or really low capacity to test this number of slots idea to see whether the high capacity subjects really rise up to 5 items and then asymptote after that. And so what I'm showing you in red and blue are those subgroups of the really high, where 5 or better, versus the 2 or better. And what you can see is that for the high group and the low group, they both rise up to 3 items. And then after 3 items, the high capacity subjects have pretty much reached asymptote. You see a little bit of a drift up, but it's not actually significantly greater than what they had for 3, not nearly as much as you have from 2 to 3. Whereas the low capacity subjects, they show exactly the same rise from 1, 2, and 3. They're identical. They're indistinguishable from extremely high capacity subjects. Yet what you see is that after 3 items, you actually see the amplitude of this activity start to monotonically decline as you increase the number of items. And the first thing I'll mention is that, well, this doesn't really fit with the number of slots idea, and that is, well, you would expect that if they have a K of 5, if they really have 5 active representations simultaneously, you would expect that, well, at least a simple model would be, you would see a continuing function up to 5, and then it would asymptote. And this actually suggests that, well, maybe one reason why they're able to perform really well is maybe they're doing something else, maybe they're grouping the items, maybe they have a better long-term memory for performance on the task. So it doesn't really fit very well with this variable slot model. Same thing with the low capacity subjects. If they have a K of only 2, well, you see the same rise. They look indistinguishable from high capacity subjects up to 3 items. And it's really when you overload capacity is when you actually see some distinction between the high and low capacity subjects. Now, to kind of square this with the data we saw before, essentially what we were looking at before was the individual differences, looking at the increase from 2 items to 4 items. And at that time, we thought we were measuring the slope of this function because we thought everyone was holding asymptote. But it turns out, now that we have a large enough sample and all the set sizes in between, really what we were actually inadvertently measuring is this drop that the low capacity subjects have. If you look at 2 versus 4, for high capacity subjects, it'll be a larger value than for the low capacity subjects, primarily because these guys, after 3 items, start to drop down. And so we were just getting the tail of that. And so that aspect of it seems to be sort of an important detail in terms of connecting this activity to behavioral performance or individual differences in capacity. Now, for a long time, we were trying to come up with these sophisticated explanations for why the activity was dropping. We came up with all these exotic explanations for it. Maybe they're clustering all the items together into one big mass, and it's a single percept, and maybe it's just like a grouping effect or something like that. But then after about 6 months of spinning our wheels, I came up with a very simple, at least, way of looking at the data that I think gives us some insight into it. And that is, well, a CDA is the difference between a contralateral activity and ipsilateral activity. And there are 2 ways that that contralateral difference wave could show a decline. You could have a decrease in the contralateral activity, or you could have an increase in the ipsilateral activity. So we went back and just simply split the data into contralateral activity versus ipsilateral activity as a function of number of items. And what I'm showing you here is these are the high and low capacity subjects, just the contralateral activity alone. What you see is that you get a rise up to 3 items and an asymptote for both subjects. And it holds consistently for high and low capacity subjects. And that necessarily means that we get this interesting effect in ipsilateral activity. If you look at the high capacity subjects, irrespective of number of items, the ipsilateral activity is pretty much a flat function, just a little bit above 0, but they're pretty much able to kind of shut off that side of the display and not really respond to it. But the low capacity subjects, what you see is that they look indistinguishable from the high capacity subjects for 1, 2, or 3 items. But after that, the ipsilateral activity starts to show this ramp up with number of items. And one way to think about that is that, well, in our displays, because we have a bilateral display, as you increase the number of items on one side, you're also increasing the amount of irrelevant information on the other side. And we think that this is consistent with the idea that, well, as you get to more and more distraction on that side, the low capacity subjects have a harder time suppressing the irrelevant hemifield, and as a result, some of that information starts to sort of come into that hemisphere and potentially even disrupt performance. So that's some recent data we've been looking at that. So at least kind of fits with some of the work that we're doing that I'll sort of focus on for the rest of the talk, looking at differences in attentional control and how individuals deal with distraction. So for this part of the talk, I'm going to sort of focus on experiments that we've been doing recently where we're asking subjects, we're measuring people's memory capacity and then putting them in attentional task situations and asking them to sort of voluntarily control their attention, and to see if there are differences in the ability to exert control over what they're going to store in memory, and how that might be one possible reason for the difference between high and low capacity subjects. So here's one experiment that we did in an early series where we're using the CDA to determine how effective someone was at controlling at what they're going to store in memory. And we gave the subjects a simple rule of just remember the red items and ignore everything else. So you queue them up to one side and they just have to remember the orientation of the red items. We had 3 primary conditions. We had 2 red items alone in the hemifield, 4 red items alone in the hemifield, and these gave us baselines for what an amplitude of 2 looked like or an amplitude of 4 looked like for that individual. And then the critical third condition that was mixed within blocks was we gave them 2 red items and 2 blue items intermixed with the irrelevant items intermixed with the blue items. And the idea is that, well, if the subject was perfectly efficient at keeping the blue items out of memory, it'd be almost as though they didn't exist, and you would expect the amplitude for here would be identical to 2 items alone. If they're perfectly good at it. If they're perfectly bad at it, that would mean that they got the blue items and the red items in memory, and thus the amplitude of the CDA here would look identical to 4 items alone. So essentially what we did was we measured people's memory capacity, then put the electrodes on, had them perform this task, and then divided them up afterwards, their data up afterwards on the basis of their memory capacity. And what we find is that, well, here are the high capacity subjects. Here, the black line is 2 items, the blue line is 4 items, the purple line is the distractor condition. The high capacity subjects are very good at keeping the irrelevant items from being stored in memory. In contrast, when you look at the low capacity subjects, you see a very different pattern. You see the purple line is identical to the 4-item line, suggesting that they're inefficiently or unnecessarily storing the irrelevant items in memory. And we're sort of arguing that sort of poor control of keeping those items out of memory sort of may actually cost them because they're storing more information, and so it makes it harder for them to accurately report the targets themselves. You can look at this, instead of just doing a median split like this on capacity, you can see that there's this nice continuous relationship between memory capacity and how efficient they were at keeping the relevant items out of memory. And this is the ERP data, just how close is the distractor condition to the 4-item versus the 2-item condition. See a nice strong correlation there. Now, more recent, what I'll also mention is that in this experiment we did it with color selection, but we've also done it with location-based selection and shape-based selection. We see the same types of results. So it's not just something that's specific to color selection. Now, more recently we've been interested in trying to understand why low-capacity subjects are poor at filtering irrelevant items, keeping them from working memory. And we initially sort of contrasted 2 possible models of that, and one is that, well, maybe the low-capacity subjects have just poor voluntary control. Well, when you give them the task instruction, maybe they're just not that good at being able to really voluntarily orienting based on color or being able to take information from the queue and actually intentionally shifting what their attentional bias is going to be. So maybe they have a weak selection signal in general. An alternative to this, which isn't necessarily mutually exclusive, but an alternative nonetheless, is that it might have something to do with involuntary orienting. Maybe instead of being poor at voluntarily orienting, maybe they're the same in their ability to do that, but that the low capacity subjects have this really strong attentional capture system or signal where distractors really capture their attention, and so they have a hard time inhibiting responses to irrelevant items in that regard. So it's more of a bottom-up type of effect, similar to Yantis and Janitas type of effects. So for example, you could have a capture signal for all items when they first onset, and it might be that the low capacity subjects have a hard time. They maybe have a really strong signal from these irrelevant items, or equally strong as they would for the targets, and as a consequence, how well you can deal with that type of attentional capture when the items initially come on the screen might determine whether those distractors were encoded and ultimately represented in working memory. And so what we did was we were interested in measuring how susceptible individuals were to different types of attentional capture. And the way we did that was, before I show you that paradigm, I'll show you some sort of basics, although many of you are probably pretty familiar with this, on how people over the years have measured spatial attention with ERPs. So there are a number of different ways of doing that, but one common way is using a bilateral display. It's something that Hayo Heinze, I think, was the first person to really do this, but Ron and Steve have done things like this over the years. And what you do is you have subjects, they're fixating, and you tell them to, say, attend to one side in advance, and then you present a bilateral display. The beauty of this design is you can have both the attended and unattended stimuli presented simultaneously, and you can look for ipsilateral and contralateral differences in the evoked responses and give you a sense of where attention was focused. And often what you find, it's not always the case, but frequently this case, is that what you find is that the P1 response, it happens within the first 70 milliseconds of the stimulus coming on. You'll get a larger contralateral P1 with respect to the side of attention. You'll often, not always, but this one is the one that varies a little bit. I'll have to ask Ron or Steve about this. But you often do find that you see an ipsilateral N1 attention effect. I mean, it could possibly be also some carryover from the P1. So when you do a subtraction between ipsi and contra in these types of tasks, what you'll often find is two positive waves, because this is if you do contra minus ipsi, you'll get this effect and then also the N1 effect that trails on it. So sometimes that's confusing because the N1 will be a positive effect. But you guys get the picture. So here's how we went ahead and, I'll mention that in these types of tasks, to see these modulations of the early visual evoked responses, at least as far as I've been able to tell, typically attention must precede the onset of the stimulus. If you shift attention after the stimulus comes on, you might pick that up in later components, but in terms of these initial visual evoked responses, typically they're primarily observed when you've already attended to that location in advance, and then the stimulus comes on and you modulate the incoming perceptual response to that stimuli. And this activity is most likely to be generated in extrastriate cortex. So we can look at the first 100 or 150 milliseconds of stimulus onset and see how attention is allocated during various attention tasks. So here's the basic task that we set up. This we refer to as the distractor capture task. And what we do here is we first give the subjects a cue, a spatially informative cue. They're fixating, and it's essentially this double diamond here. And on each diamond, there's a little red, a colored square that's somewhere on one angle, a corner of those diamonds. And so one group of subjects will be told, okay, you'll pay attention to red cues. The other group will do green cues, so we counterbalance that. And this cue, it looks kind of complicated, but essentially it gives them it tells them with 100% validity where the target will be on the upcoming trial. And so in this situation, this little diamond is a schematic of what they're going to see. It's a little diamond configuration of Land sees. And so a red dot on this corner means that this is the valid location. This is the target location. Does that make sense? Okay, so they'll just go ahead and their task will be, when this array comes up, and it'll just be on for 50 milliseconds, they have to report where the gap was in the object that was presented at the target location. And that's it. That's the entirety of their task, what they have to report. And so we ask them to sort of focus on their accuracy for being able to do that. Now, by time locking the ERPs to the onset of this target array, that gives us a good measure, I would argue, of their ability, each subject's ability to take in information from this cue, which is relatively complex cue, and voluntarily orient their attention to a target location. So it would give us a real good measure of how well they could take that task instruction and voluntarily orient spatial attention prior to the array coming on. However, if attention is captured by these distractors, well, that would happen after those stimuli came on the screen. And so their ability to orient, and when we time lock here, that's going to tell us how well they could orient before the thing came on. But if they reoriented attention afterwards because of the distractors, well, we wouldn't really capture that by time locking to this. And because of that, that's why we had task-irrelevant probes that quickly followed the target array. So 50 milliseconds After the target array, we would flash a probe, and it would be a bilateral one, just so we could look at this contralateral ipsilateral difference. And we could either flash the probe at the target position, or we could flash the probe at one of the distractor positions. Or on some trials, we would have no probe altogether. And that allows us to separate out. We could do the probe versus no probe for the ERP aficionados out there. That'd be a way for us to isolate activity to the probe and separate that from all the overlapping activity from the target array that will last for a few hundred milliseconds. So we can look at the responses to the probe. And here we're going to argue that this gives us a measure of how they may have changed the spatial allocation of attention because of the presence of those distractors. So here's a little cartoon of how that might occur. Say, for example, if you have this little purple spot, maybe this is the focus of attention on the target location before the target array appeared. So they got the cue and they shifted over there, and they're got a nice tight focus of attention at the target position. And imagine you have an individual that has perfect resistance to attentional capture by the distractors. When you flash the array on, including the distractors, they hold their focus really well just on that position. And now, moments later, if you probe outside of the focus of attention, you won't get much of a response to that probe because it's not where they're attending, so we'll get negligible P1 response. But if you flash a probe at the target position, that's where they're holding their focus, you should get a nice big P1 attention effect to a probe there. And that would be an individual that could hold their focus really well. And you can look at the difference between the probe at distractor and probe at target, and that would give you a sense of how well they could hold their focus. There would be a nice large difference between probe at target, probe at distractor. In contrast, if you had an individual that was really susceptible to attentional capture by the distractors, maybe they start off with the same attentional focus, but that when the distractors appear, they either broaden their focus, or another possibility is that the presence of these things just pulls the attention to one spot or another spot. It could either be a broadening or kind of shifting it to another position. I think but the predictions are the same, and that is if you flash a probe now at a distractor position, you might actually get a little bit of a response because they may be attending there as well. Or if you probe at the target position, you'll get a reduced response because they may have had their attention pulled off of just the target position. The bottom line here, though, is that if they're susceptible to capture, is that there would be a small difference in the P1 response to the probes in terms of the gradient between probe on target and probe on distractor. There'd be a smaller difference as opposed to someone who could hold their focus really well, which would predict a much larger difference. So what I'm showing you here is the P1 attention effects time locked to the target arrays, which I said before, I was arguing that tells us something about their ability to take in the information from the cue and shift attention voluntarily over to that target array. And what you can see is that we get nice big P1 and N1 attention effects. They're on the order of, I didn't show the scale here, these are about almost a half a microvolt, which sounds small, but for ERP attention researchers are like, oh, that sounds about right, more or less. It's not a millivolt, it's half a microvolt. But you see a nice normal looking one equally for the high and the low capacity subjects. They're about the same. If you measure the magnitude of the amplitude of the P1 attention effect and you plot that as a function of memory capacity, you really don't see any correlation at all. Pretty much they can both equally orient their attention over and modulate the incoming perceptual response. So they're not impaired in that regard. However, if we look 50 milliseconds later, 50 milliseconds after we see where there's no differences, we just look at the response to the probe, which might tell us something about attentional capture, there we see huge differences between the two groups. So here what I'm showing you here is the attention effects to the probes. Here we have to do the contralateral minus ipsilateral difference waves, because we also do this extra subtraction of subtracting out the no probe trials. Sounds like a lot, but I'd be happy to walk through if people have questions. But here are the high capacity subjects for the probes on target versus the probes on distractor. And what you see is that when it's a probe on distractor, for the high capacity subjects, you pretty much get nothing. You get pretty much a flat line when you do the contra minus ipsi, meaning that there's not really much of a difference between the attended side and unattended side for probes at distractor, which makes sense. They're holding their focus. They're not attending their distractor positions. But when you probe on the target, you get these big, clear P1 and N1 attention effects. They're nice and large, and there's a real big difference between probe on distractor and probe on target. You see a big effect for the high capacity subjects. But for the low capacity subjects, here where you see a much more substantial difference. And that is, well, there's two main things. One is that in terms of the response to the target, it's much smaller. And it's not just that these guys have smaller P1 attention effects in general, because moments earlier we saw that they're exactly the same. It's considerably smaller, but you also see that that the response, you'll have to kind of squint a little bit, but the response is actually significantly greater than 0 for probes on the distractor. And in later experiments, we found that it actually had to do with the fact that there's 3 distractors, and we were just randomly probing one of them. So we had to get lucky that they happened to be attending that particular one. Later on, we would either have fewer distractors or probe multiple distractors, and you can actually see a big response for the low capacity subjects when you probe the distractor. But the real key here is that there's a very small difference between the probe response to distractor and target for the low capacity subjects, whereas the high capacity subjects showed a much greater difference between the two. And what we find, if you can just look at this as a bar graph, you can see a really large interaction between capacity group and the position of the probe, which sort of fits with this general idea that the low capacity subjects show a greater susceptibility to attentional capture. We can look at this in terms of correlation. We can just simply look at the P1 effect, looking at the target minus distractor difference as a function of memory capacity, and we get a really nice linear correlation between memory capacity and this susceptibility to attentional capture, at least that's what we're calling it. And this is a recent study that we published in Journal of Neuroscience. So we get a real strong correlation between capture and memory capacity. And in subsequent experiments, we went through and we've replicated this effect multiple times, but we've gone through to see, well, do you see this, is this something that's specific to these distractors, or is it something that's just more general about their responses to probes? So we did another experiment where we either had the same thing as what we did before, where we had a target and very similar distractors, or we had highly dissimilar distractors. It could be that the low capacity subjects sort of reallocate their attention anytime there's anything, even if it has nothing to do with the target they're looking at. Or we even had when there was no distractors at all to see if without capture maybe you shouldn't see the effect at all. And what we find is that when there's similar distractors, we replicate the general effect. When there's dissimilar distractors, the effect goes away altogether. When there's no distractors at all, again, we see no effect. So really, we see this general relationship when the distractors and the target share a lot of features. They're very similar. And this would be something akin to what's referred to as contingent capture, like Chip Folk and Howard Eguth have been talking about over the years, where you have attentional capture by distractors that partially match what the current attentional control settings are. And that seems to be really what we're hitting on with these low capacity individuals. Now, in terms of attentional capture and memory capacity, there's really two possibilities in terms of explaining that relationship. And one possibility, and that I was kind of talking about before, at least our original data suggested that it's about susceptibility, that the low capacity subjects are more likely to be captured than high capacity individuals, at least in these contingent capture situations. Although there is an alternative, and that is, well, it might be really more about disengagement time. And that is, well, maybe everyone has equal susceptibility to attentional capture in those situations, but the low capacity subjects take longer to sort of disengage from that irrelevant item, and as a consequence of kind of hanging on to it too long, they end up encoding that item into memory, and that starts a whole cascade of problems for later on, where the high capacity subjects might be able to unhook quickly and get back to the relevant task. And that might be a reason why we saw these effects. So in this experiment, what we did was, this is a behavioral experiment where we just wanted to show in a different situation, can we see attentional capture effects? And we also wanted to measure the release time or the disengagement time from attentional capture. And the way we did that was we first gave the subjects these placeholders. And these placeholders they're 4 boxes, and they stayed on the screen the whole time. And we told the subjects the task-relevant stimuli that they're going to see are only going to ever appear within those boxes. Anything other than outside of those boxes is totally irrelevant, and you can ignore it. And what we did was those boxes were there on the screen, and at some point, some Landolt Cs would flash up inside those boxes, and they were told, okay, we want you to tell us, it's essentially a quick visual search task, and they have to tell us, we told them, okay, the targets are red for you, or another group had green targets, and they had to just report where the gap was in the red box, in the red Landolt C. And then these items were on the screen for a short amount of time. We actually staircased this for each subject, without any flankers, to sort of get their sort of at a particular level of performance. I think they were at like 75%. And then we go ahead and mask them to keep performance away from ceiling. Now, what we did, though, it's sort of that's the basic task. What we did was sometime prior to the onset of this display, we flash a task-irrelevant flanker. So a colored box outside of one of these placeholders. So they know it's totally task-irrelevant, and so we can either flash a box that was the same color as what they're searching for. That would be a sort of a contingent capture type of a flanker, something that would be sort of partially match what they're looking for, so really be disruptive. Or sometimes we would flash a yellow box that was a different color from anything that they'd seen on the screen. And so this is a different form of attentional capture, similar to like what Jan Theovis has been studying, looking at sort of or Singleton or pop-out capture, where you have something that's not related to the task you're performing necessarily, but it's really more of something really salient or surprising that comes up on the display, and maybe that also sort of captures attention. And so what we did was we had either a contingent capture or a Singleton type capture event happen, or sometimes there was none at all. And we would vary when that happened relative to when this came on. So we'd vary the SOA or the timing between that item and when the target array came on, so we could measure how long it took people to sort of recover from the capturing event that came on. And so what I'm showing you here are the capture effects for the Singleton capture conditions. And essentially what we're measuring capture effect is we look at distractors absent minus present. So essentially how much of a hit did they take when that particular distractor came on or that particular flanker came on. For Singleton capture, and so what I'm plotting here is the SOA. So this was a 50 millisecond SOA, 50 milliseconds before the target array, 150 milliseconds up to 350 milliseconds. And the yellow line is the low capacity subjects and the red line is the green, the high capacity subjects. What you see is that at the shortest SOA, you see about an 8% decline in performance because of the presence of that Singleton capture. But it's equivalent for high and low capacity subjects. But that when you go to 150 milliseconds, both groups have already kind of recovered back down to within 2% of distractor absent, and it pretty much is stable. And there's really no difference between high and low capacity subjects in responding in the susceptibility or the recovery time for Singleton type capture for high and low capacity subjects. They're equivalent. However, in the contingent capture situation, we saw something a bit different. And that is, well, at 50 milliseconds, high and low capacity subjects show equal amounts of disruption in their performance. They show around a 10% decline in performance because of that contingent capture item. However, when you go to 150 millisecond SOA, you can see the high capacity subjects are already really recovering. They're down now about 4% of disruption, and by the time they get to 250, they're almost completely recovered entirely, whereas the low capacity subjects are equally impaired at 50 and 150, but then they don't actually start to show recovery until about 350 milliseconds. So we get this real interaction between capacity group and the SOA between the distractor, the contingent capture distractor, and when the target array appears, suggesting that these low capacity subjects take longer to recover from a contingent capture. And what we did was we wanted to try to come up with an individual estimate of recovery time for each subject in this. And one at least simple approach that we've taken so far is to essentially make a threshold at 5% to find, okay, well, what time point did each subject recover to the level of about 5% or better of their distractor absent condition. And what we find is that we can come up with this release time measure for each subject and see that we get a nice correlation, a negative correlation with memory capacity. High capacity subjects show longer release times, and this declines as you increase memory capacity, or the high capacity subjects have a very short release time. So at first blush, this data appears to be inconsistent with our previous experiment, the ERP experiment, where we showed that it looks like the high capacity subjects are really able to resist capture and the low capacity subjects are really susceptible to capture, at least in terms of the ERPs. But here it appears that they're equally susceptible to capture. It's really the difference is about the recovery time, how quickly they can disengage and get back to the task. So, but one possibility in that experiment, in the ERP experiment I showed you before, was we use a single SOA for the probe. We used a 100 millisecond SOA between the target array and the probe, and then we just used that for a fixed number. And it could have been that we just happened to hit right somewhere in the middle of that recovery time, where there's a maximal difference between high and low capacity subjects. So essentially what we did is we went back and did that experiment again, and we varied the SOA with the probe. So we either have we had 3 target to probe SOAs. We had 50 millisecond, where the ray comes on, and as soon as it disappears, a probe appears. So it's immediate. So that'd be it's a bit shorter than what we used before. Or we had a 100 millisecond one that's identical to what we used before. And then we also had a longer one that was 200 milliseconds. So to see, like, they sort of go even further away. And so essentially the same basic logic appears, and I'll go ahead and show you the results from this, looking at the probe responses. So what I'll show you first are the group data, all subjects, irrespective of memory capacity, for probes on target and probes on distractor as a function of the 3 SOAs. And what you find is that this is the probe at target and this is the probe at distractor for the 50 millisecond SOA. And what you see is that even at the group level, there's a smaller difference between these two lines in this time window, in the P1N1 time window. But then when you go to 100 milliseconds, you see a much larger, you start to see a large response at target versus distractor, and that pretty much holds, and maybe is even a little bit bigger when you get to 200 milliseconds. That's for all subjects. You can measure this just looking at mean amplitude. And essentially, this is the target line, this is the distractor line. There's a small difference between these two values at 50 milliseconds, suggesting you're getting a fair amount of attentional capture, that there's not much of a disparity between probe at target and probe at distractor. But then when you give them a little extra time, you actually see a big spread out, and it pretty much reaches an asymptote at 100 milliseconds. And you can look at the difference between these two, just as I talked about before. We can just do a subtraction between these two lines, and that's what this purple line is here. That's what we'd call this P1N1 capture effect, where you see the smaller numbers are more capture, the larger numbers are less capture. So at the shortest SOA, you get a large amount of capture, and then pretty much you see some recovery at 100 milliseconds. Now we'll go ahead and show you these functions, these capture effect functions for the high and low capacity subjects across the 3 SOAs. And what you see is that, it's kind of hard to see, sorry, wrong colors choice, I think. The green lines are the high capacity subjects and the yellow lines are the low capacity subjects. And what you can see up here at 50 milliseconds is that there's really not much of a difference between the high and low capacity subjects when you look at a probe immediately after the display comes on, 50 millisecond SOA, pretty much everyone's showing a capture effect. It's a small effect, and there's really no difference between high and low capacity subjects. But when you get 100 milliseconds, now suddenly there's a separation between the high capacity subjects are recovering and the low capacity subjects aren't. And then by 200 milliseconds, now everyone is back to the same. High and low capacity subjects are indistinguishable when you go to a 200 millisecond SOA. And you can look at this as bar graph. It's a little easier to see. This is the P1 and capture effect as a function of SOA, and you can see that at 50 milliseconds, high and low are the same. They show they're not significantly different at 50 milliseconds, but at 100 milliseconds, the high capacity subjects are really recovered, where the low capacity subjects are just showing a little bit of recovery, but not nearly the same level. But then by 200 milliseconds, they've converged again. So both groups have recovered. Now, one thing that we did in this experiment was, in addition to this ERP experiment, we also ran that same behavioral measure that I showed you before of the release time. So we actually had 2 separate measures of release time. We wanted to see whether they both tapped into the same underlying construct. And so what we did was we measured some of the release time on the basis of that behavioral experiment, and then saw how well it predicted their data in this ERP task. So, before I show you that, here's just the correlation of, this is just a replication of the original experiment. So if we just look at the 100 millisecond SOA as a function of memory capacity, we see a nice correlation as we had in the previous experiments. Now I'll show you how we relate the behavioral release time measure with this ERP release time measure. And here is, essentially in this one what we did was, this is the ERP effect. And what we did was we took that release time measure, the behavioral release time measure, and we just divided people into fast releasers and slow releasers, irrespective of K, although it does correlate with K. But just see how well that split would sort of explain the ERP data we find. So these are the fast releasers and the slow releasers. And we look at this P1 response, and at 50 milliseconds, there's not much of a difference between the groups in terms of the P1N1 capture, but there's a big one at 100 milliseconds, and they go back and converge. So we do see a correspondence between those two. We can also look at, this is the release time estimate for each subject based off of that behavioral task. And this is the P1N1 difference at 100 millisecond SOA, where we see the maximal difference between the two. And we see a nice, strong correlation. Individuals with a really short release time show a real good recovery from capture at 100 milliseconds, whereas the slow releasers, which are also the low capacity subjects, show still at 100 milliseconds that they show a lot of capture, that they haven't recovered yet. Okay, no, that's a lot to digest, but I'm pretty much done. So essentially what we find is that overall, across these different measures, it appears that low capacity subjects, they're not impaired in terms of their ability to voluntarily orient attention. It doesn't appear that they have a reduced number of slots for holding items, at least up to 3 items. They seem to be indistinguishable from high capacity subjects. But really it seems to sort of emphasize this, something about attentional capture, and in particular how they recover from attentional capture. So I know you've been patient with me with a lot of words I've thrown at you, so I'll keep my conclusions fairly short. Memory capacity strongly predicts the efficiency of attentional control over working memory storage. We see strong covariation between memory capacity estimates and how effectively someone can control what's going to be stored in memory. Individual differences in filtering efficiency might be the result of the ability to resist attentional capture by distractor onsets, or possibly even at least recovering from attentional capture by distractor onsets. And this capture appears to be only in situations when it's a contingent capture situation, when the distractors partially overlap in terms of features with the targets that they're looking for on that particular task. You don't see it that it's not as though any sort of surprising or salient event that doesn't overlap with what they're looking for. The high and low capacity subjects are indistinguishable in both their susceptibility and also the recovery time from Singleton capture. The initial selection by capture may be the vehicle for unnecessary storage of irrelevant items in working memory. We did an experiment, I didn't have a chance to talk about it today, where we actually looked at the CDA in terms of filtering, and it was a Frankenstein experiment combining this P1 probe response looking at capture and also looking at the CDA for whether or not the distractors ultimately got stored in memory. And we see this really nice correlation between those two. If you showed a real capture effect at P1, then you are more likely to have stored those irrelevant items in memory. That's in that Journal of Neuroscience paper we just published. And then finally, the high and low capacity subjects are equally susceptible to contingent capture and other forms of capture, but the low capacity subjects really appear to take longer to disengage following a capture, and as a consequence, they might encode those items unnecessarily, and that might cause this entire cascade of problems that ultimately might even sort of explain why they sort of have a hard time dealing with different types of aptitude measures and other types of structured attentional and educational settings where they often sort of have failures. So I'll go ahead and stop now, and I'll just, the last thing I'll say is I'd like to thank my students who did all the work, particularly Keisuke Fukuda and Andrew McCullough, and this work was funded by NSF, and I'd like to thank you for your time and attention. Yes, here in the front.
You pointed out at the beginning that there's a strong correlation between these visual memory aptitudes and general IQ. And if you take a kind of traditional view of working memory, you can sort of tell a story like fairly compelling or intuitive, that the more you have in working memory, the more you can access information that leads to intelligent decisions.
Definitely
and so on.
have more things to contemplate at the same time.
Exactly. But now, with the alternative story you're telling, where it's an attentional issue, and it's even a temporary attentional issue, the explanation for that correlation becomes much more difficult. And I just wondered how you get to explain the correlation with this kind of fascinating and very compelling set of data that you've given us, showing that it's a lot more complex than just the size of item.
Right, exactly. That's an excellent point. I mean, I think that was sort of underscores, many years of work, assuming that, more information in memory at the same time allows you to make these more complex associative relationships. And that might ultimately explain intelligence. And while that may be the case, and I guess we can't completely rule that out entirely, although so far we at least have this attention data that I think is compelling, a lot of it might actually be how, in terms of these structured tasks, like when you have someone do a standardized test or these different intelligence tests, a lot of success and performance on that is not necessarily contemplating more and more items, but really essentially being able to exclude irrelevant information that's there embedded in what they're looking at. And I think stronger attentional control or having less information actively represented in memory in some ways might actually be really an advantage. So for example, even when, even outside an aptitude testing measure, you can think of, in the real world, if you think of average memory capacity somewhere around 3 items, well, just about any environment that you're in, you're going to have far more objects in view than you can actually represent in memory. And we have this sense that we're able to sort of hold all this information in awareness, but actually when you test it, it's actually really limited. And if you think about it, well, having an extra 1 or 2 items in memory might be advantageous to some degree, but often it's not going to really help if you have a poor control over what gets into memory. So this is kind of where one metaphor we've used over the last few years is really, it's not how big your nightclub is, it's how good your bouncer is. And if you've got, a really small nightclub, but a really good bouncer controlling who gets in, you keep the riffraff out, and you're going to have only the really relevant information that can be represented. And as opposed to having a larger nightclub and a say, drunk bouncer, I don't know how far I'll go with this metaphor, but you just sort of who lets just anybody in, well, you can quickly overload, even if you had more space, you could quickly overload that, and you'd sort of be mixing in the really relevant information that was specific to the task you're engaging in, along with the irrelevant information that may ultimately sort of end up costing you. And so that is definitely a good point that I think there's still a lot of work to be done to really establish that, but I think that having a tighter control and having less information in memory in many ways might ultimately be more advantageous because you can just focus on the really relevant work. And one thing I'll just mention real briefly is that it's not the case that low capacity subjects are just bad at everything, right? Because they're actually pretty unimpaired and normal in many situations. And actually, and one question that we've had over the years is, are there situations where they're better? They have an advantage as opposed to high capacity individuals. And when you have a structured testing situation like an IQ test or like a reasoning task and things like that, well, they're going to have a disadvantage there. But maybe there's some advantage to having more information or sort of more irrelevant items that get information that gets into working memory. Maybe that's advantageous in creating sort of novel associations. And that was actually something that I'm collaborating with my colleague Marjorie Taylor at the University of Oregon, who studies creativity and imagination. And what we find, at least in one of our preliminary studies, is that low capacity individuals are really good at being able to generate novel creative associations between things. They score higher on creativity measures than high capacity subjects. And you actually get this sort of there is possibly an advantage when you take people out of a structured testing situation where they have to focus on this stuff and ignore all this other stuff. If you take them out of that, there might actually be an advantage for sort of letting irrelevant information to flood in. Yeah, Peter.
So I'm wondering what's going on in the front of the brain during all of this.
Yeah.
And whether or not you've looked at frontoparietal interactions and
That is
if there's anything that tracks the...
That's a great question. I have a slide on that. We haven't done it directly, though others have, and I'm grateful for them. There was this great study by McNabb and Klingberg came out about a year and a half ago, where they were essentially interested in the same issue. Because essentially what we're looking at, particularly with the CDA, is we're looking at sort of the consequences, the ultimate consequences of how effective someone was at controlling their attention. We can just see what ultimately ended up in working memory. But a lot of these attentional control is often thought of being more prefrontal cortex. And so we're just looking at that sort of ultimately what gets there, but it could be that a lot of the action is really happening sort of in frontal cortex. And so what these guys were doing is they essentially did the same task as ours, where you remember the red ones and ignore the yellow ones in a memory task. But what they did was the kind of clever manipulation was that before the memory test came on, they gave them a cue. There was just a central cue, it was either a square or a triangle, that told them whether or not they're going to have to filter on the upcoming trial. So in ours, they didn't know until they saw the display. In fact, most of the time it was actually no filtering, and then sometimes it would kind of surprise them because 1/3 of trials had filter. But here what they did is they pre-cued them, told them, okay, square means they're going to have to filter the yellow ones. A triangle means just go ahead and remember everything that comes up on the screen. And what they found, and that was like a few seconds before the memory array, and so they could just time lock that activity using imaging to see what areas show a differentiation between these different types of filtering cues. And what they found was that in the basal ganglia and also in the prefrontal cortex, they saw that there was what they refer to as filtering set activity, where there's a ramp up of activity in certain cortical and subcortical areas for filter cues relative to no filter cues. In particular, in the globus pallidus in the basal ganglia, they found that this ramp up for when they're doing filtering relative to no filtering really nicely predicted memory capacity. They saw that the high capacity subjects really show this ramp up in basal ganglia when they're expecting to filter relative to not filtering, versus the low capacity subjects, which really didn't show much of a differentiation between the different types of cues. Then what was actually pretty cool with this study was that they looked at this filtering set activity, and they looked at the activity when they're doing the memory task, like during the retention interval. And they could measure in parietal cortex, so in posterior parietal cortex and in the vicinity of IPS, they could essentially come up with this irrelevant storage measure similar to ours, where you see distractors present, targets plus distractors. Does that have more activity than targets alone? It's similar to the same logic as what we found. They found exactly the same thing we did, so we're happy they replicated us with imaging. But what was particularly cool was that they were able to link up this filtering activity in the basal ganglia prior to the trial starting and this unnecessary storage effect. So this is kind of that, it's not the direct interaction, but I think it kind of insinuates the interaction, where if you really engaged in this filtering set activity, you had a large amount of filtering set activity in the basal ganglia, that predicted that you would have very little unnecessary storage in parietal cortex. But if you had very little of that activity, then you showed a large unnecessary storage. And so I think that might get at some of this general neural model of connections between these frontal and even like subcortical structures like the basal ganglia and what you ultimately store in memory. Yeah, Joy.
One more question.
Yeah.
So I just have you showed us really interesting individual differences in intentional control.
Mm-hmm.
And you sort of divided that information up in terms of low and high visual working memory capacity. So my question is there something special about visual short-term working memory capacity per se, as opposed to, say, verbal working memory capacity?
Right.
Or is it just that there are individual differences in attentional control, and that these things are giving rise to what you are measuring as visual short-term working memory differences?
Yeah, that's a great question. I'd say there's a couple of different answers to that. One is that I would say that when we're looking at working memory performance in this task, when we look at average capacity, that is actually telling us something about working memory capacity specifically, and that the variance around 3 or so items, going back to my original grad school work, the variance across individuals is probably something about attention, but that the core capacity is pretty much that. Now, you had asked about do other working memory measures, particularly like verbal, show similar types of measures. And well, if you use a simple version of a verbal memory task, like a digit span task, where you just give them a sequence of numbers, you can get hints of these types of relationships. But often it's obscured by the fact that there's a lot of different strategic approaches to doing digit span tasks. You can have different chunking effects, and then you have also different abilities to verbalize information. Like people who can articulate really fast often can sort of hold more information. And so it often is tricky to get that just with a very simple visual or verbal memory storage measure. So what many people do in the literature is come up with these more complex span measures, where they'll combine a measure like that with some additional task. And so, for example, operation span is one that is particularly popular. Reading span was the original one that Daneman Carpenter did. But essentially what you do is you interleave two different tasks where it's you give them, for example, an operation span, you give someone a simple word math problem like, is 5 plus 2 times 3 greater than 10? Right? Something that they can figure out on their head, just in their head, but it makes them sort of hold that information temporarily in working memory, and they'll say yes or no, and then they'll give them a word, giraffe. Then they'll give them another math problem, and then another word, and then another math problem, another word. What that really does is it sort of disrupts their ability to use either chunking or verbalization of those words, and then at the end you just test them on their words. And in those situations, you actually do see very similar types of effects, like Mike Kane and Randy Engel and Nash Unsworth in their general group have found a nice set of relationships that are very similar to the kinds of effects that we've found here. Now, I would argue our task is better because I'm arrogant and biased, but I think it's true, though, and that is the arrogant and biased part. And it's particularly because in those tasks, there's a lot of moving parts because you've got this dual task situation. They have to switch back and forth. And often you get weaker relationships with attention, partly because there's multiple sources of variance in the behavioral measure of what they call working memory span. In our task, it's a little bit more of a simplistic, kind of streamlined measure, because you kind of remember the items or you didn't, and because they have a brief display, they don't have as much opportunity to engage in as much strategic approach to performing the task. So I think so. The short answer is yes, you can see very similar relationships. They tend to be a little bit weaker, I think, partially because those span measures tend to be there tends to be multiple sources of variance, and I think that you get a little bit weaker of a relationship, but you can still see the same general patterns with their work.
We better end there.
Okay.
Thank you very much.